Okay, very nice. Now next, to ammonia gets converted into urea by a cycle called ornithine cycle or urea cycle. And this cycle takes place mainly in liver. Clear? Did you understood today? So today we'll be discussing about ornithine cycle, urea cycle, or Krebs cycle. Any difficulties there? Okay. Write down ornithine cycle or Krebs cycle. Two molecules two molecules of ammonia plus one molecule of carbon dioxide will give you urea. Are you getting the point? During this process, 3 ATP molecule is used up. 3 ATP. During the conversion of ammonia to urea, there is urea, right? It occurs in liver cell, it has 5 stages. 5 stages. First stage is formation of carbamyl phosphate. Second stage B, formation of citrulline. C, can you all see the screen? Formation of arginosuccinic acid. Can you all see the screen? T. Okay. Kim, did you send the notes? Yeah, I was asking who sent the notes. Okay. The reason I asked is why did you send the notes in the Teams on the WhatsApp group? Arginine E is urea and Kim ornithine. So these are the 5 steps that takes. See, without asking my permission, you're not going to send any notes to anyone. Is it clear, Kim? I want each and everybody to write their own notes. And you, without permission, if the notes were required, I would have asked Ma'am Fina or somebody to upload it on the Teams. And you send the Teams into the WhatsApp group without asking me. You didn't inform me. Okay, and this is for everybody without asking me. Do not upload anything related to zoology. Clear? If it's concerned to a subject point, rest all it's up, it's your wish. But when it comes to the notes or when it comes to the zoology content, please take a concern. If you start uploading like this, the students out there will not write any notes. They will wait for your notes. Do you understand? The notes that were required about structural organization and about what cockroach, that was uploaded. Now Kim has uploaded digestion absorption chapter. When is your next test? The next test is in the next week, Sunday, right? So please go through this chapter of digestion absorption. It's okay, Kim. It's okay, but I just wanted to, so that you know it. Like, you do not do that because otherwise the students will not write the notes. I want each and everybody to write the notes. Clear? There's nothing wrong, but just I will, if you would have informed me, it would be better. That's it. So the next thing is upcoming test will be the digestion absorption. Now, since Kim has already posted the notes, please read through it, go through it, understand it, and if you find, this is for all the new students who have joined. If you find anything, I think she has covered all the points which I have given her, given during the class. So you can go through the notes, read it, and if you find any difficulties, you text me. Okay, text me on Teams. Is it clear? All the new pitch students, is it clear? Will you do that so that we can cover the portion that was left? If you read, you understand, you don't understand, mark it, keep the question and ask me. But I want you all to answer the test. Gargi, is it clear? Okay, well, next. So there are different steps in the Ornithine cycle. So let us study these steps. First is, we'll write 1 ammonia plus 1 carbon dioxide will give you carbamyl phosphate. Carbamyl phosphate will give you citrulline. Citrulline will give arginosuccinic acid. From arginosuccinic acid, you get arginine. From arginine, you get ornithine. Ornithine. Okay. Now, which are the enzymes? The enzymes for carboxyl is carbamyl phosphate synthetase enzyme. Here, then, here it is ornithine, carbamyl transferase enzyme. Next, there is addition of a molecule called aspartate. The enzyme here is, write down a different color here, arginosuccinate synthesis. Arginosuccinate synthetase, arginosuccinase, enzyme. Here the enzyme is arginase. Okay. Next, ATP. Let us add up the ATPs. Yeah. 2 ATP is used up here. Altogether 3 ATPs, right? So 2 ATP is used up to give 2 ADP plus 2 IP. Then here, 1 ATP is used. ATP to give plus AMP. So 3 ATPs. The one here, 1 ammonia is used. And then 1 ammonia molecule is coming from this donor. Aspartate is an amino donor. The other, I told you 2 ammonia will combine with 1 oxygen. Here first 1 ammonia is combining and then 1 more amino donor which is coming from aspartate. So 2 ammonia, presence of 1 carbon dioxide gives you urea. Where is the urea coming out? The urea is coming out here. Urea is coming out here. Urea. You get ornithine and urea. Ornithine, it are done with the same color. This is urea. It's a monophosphate. Sorry. Sorry. Cannot see the screen? Are you there? Can you see the screen now? Can you see it now? Okay, I write down. Once you're done, do let me know. I'll explain you. Hello. Why are you calling me 100 times? डेली क्लास नहीं मेरी मर्जी ओके ऐप ओके मैं अभी खाऊंगी थोड़ी देर में पढ़ी नहीं है ना ऑफिस क्लास चालू है चलो I'm keeping now अब I'm taking class now can I keep तुम 10 बार कॉल मत करो यार next done okay so ornithine cycle is the one that helps in taking care or the conversion of ammonia to urea how many molecules of ammonia to ammonia converted to form urea is it clear it's a cyclic process in the cyclic process why it is called cyclic let us study and this takes place in the liver cells half takes place in the mitochondria how much until here okay until here this part is under mitochondria and this part is under cytoplasm of the cell are you getting the point this the other part is in the mitochondria and the other part is in the cytoplasm of the cell of which cell liver cell so what happened one ammonia converts with one carbon dioxide it in the presence of carbamyl phosphate synthetase, it synthesizes carbamyl phosphate with the usage of two ATPs. Initially, what is the reaction? Two ammonia, one carbon dioxide, three ATP, right? Initially, it uses two ATPs to form carbamyl phosphate. That's the first formation of carbamyl phosphate. This carbamyl phosphate converts into citrulline in the presence of ornithine carbamyl transferase. Right? The formation of citrulline happens in the presence of enzyme ornithine carbonyl transferase. The carbamyl phosphate combines with ornithine. Along with this, there is a conversion of leading to formation of citrulline. Now, this citrulline, on the step of citrulline, there is one more ATP molecule that is used. This ATP molecule, when it is used, there is an aspartate molecule that is added up, which gives a one more ammonia, that is amino donor, which is aspartate is acting like an amino donor. Important question. In the presence of arginosuccinate synthetase, citrulline converts into arginosuccinic acid. Arginic succinic acid breakdowns into arginine and fumarate in the presence of enzyme arginosuccinase. Now this arginine converts into urea and ornithine in the presence of enzyme arginase. The urea is thrown out, whereas ornithine is going back to the cycle again. So it is a cyclic process. The name ornithine cycle or urea cycle or Krebs-Henselin cycle are the names of this cycle. So two ammonia combine with carbon dioxide to give ammonia by the usage of how many ATP molecule? Very important question, 3 ATP molecule. Any doubts to anyone? Any doubts to anyone? Aspartate amino donor, arginase, cytoplasm of the seed. This cycle happens. This cycles happens at liver cells. One is in the mitochondria, other is in cytoplasm. Right? Liver cell, half part happening in the mitochondria, other part is taking place in mitochondria. Here you have two ATPs converting into ADP molecules, and then two IP. Here what happened? One IP is released, so it become monophosphate. Here one IP is released. One IP is released here. Any other doubts? Please write down. Okay, let's move to the next. Note, very important, many times MCQs from this part. Maximum urea is found in, tell me, hepatic vein or renal vein? Maximum urea is found in dash vein. Hepatic vein or renal vein? Very good. It is in the hepatic vein, right? Liver is a site for the formation of urea. So you find it in the hepatic vein. And minimum urea is found in, come on, tell me, minimum, very important questions. Minimum urea is found in dash vein. Minimum urea is found in dash vein. Very good, Tanvi. It's a renal vein. Renal vein, right? Any doubts? Any doubts? Any doubts? Can we move to the next? The next topic is osmoregulators and osmoconformers. Osmoregulators are animals which can maintain osmolarity of the body. Irrespective of outside surroundings. Clear? Here, the animals which maintain the osmolarity of the body, irrespective of any osmolarity that is present in the surrounding medium. Those are called osmoregulators. Is it clear? Then, importance are the examples. Important examples. All vertebrates, all all vertebrates fishes, amphibians, reptiles, apes, mammals. We maintain our osmolarity of a body irrespective of the surrounding osmolarity. We don't keep on changing, right? Next, but very important exceptions, except hagfish. A very important question. Except hagfish. Yes, all vertebrates. Most of freshwater fish, freshwater invertebrates, and most of freshwater invertebrates. So you have osmoregulators, then examples for osmoregulators. Any doubts? Okay, 2 minutes. Yeah, write down.
Ma'am, most of freshwater invertebrates are examples or it is an exception?
No, it's not an exception. It's the second point.
Okay, thank you.
Which is a freshwater polyferal? Freshwater, example for freshwaters, tell me. Spongilla, yes. So this all comes under osmoregulators. Is it clear? Because they maintain the regulatory osmoregulators. Whatever the osmoregulator outside, it's not going to affect their inside osmolarity. Clear? Next. What is osmoconformers? Next is osmo. Write down. Osmoconformers. They change the body fluid's osmolarity. They change body fluid osmolarity with change in the osmolarity of surrounding medium. Example, hagfish, then all marine invertebrates, and some of the freshwater invertebrates. See, the question will be on hagfish. MCQ question will be on hagfish. Very important. Next. Come on, tell me any doubts. Any doubts? Hello, क्या हुआ क्लास में? हेलो. Okay, next. Osmoregulatory roles of kidney. Osmoregulatory role of kidney. The first condition is when body fluids. How is this? We know that kidney not only uh, excrete the waste out of the body, but also maintain the osmoregulatory or osmolarity of the body. So how does it do? So let us study about this. First, when the body fluid or the volume is increased. First condition, when body fluid volume is increased, or when increase in blood pressure, or due to excessive water intake, or this happens due to excessive water intake. What the kidney will do when there's an increase in, so when there's an increase in body volume, there's a pressure on the arteries, right? So obviously the blood pressure increases, one of the reason. Now, what should be the aim of the kidney? To produce hypertonic or hypotonic. So when the condition is like this, what is the aim of the kidney? When the body volume is increased, what should be the condition of urine? Hypertonic or hypotonic? Very good, Chrisley. The urine should be hypotonic. So we need to excrete the urine that is hypotonic. This can be achieved by increasing the ultrafiltration. One method is by increasing in ultrafiltration. By increasing ultrafiltration, how? First thing is increase in blood volume increases the blood pressure, which when the blood pressure is increasing, there's an increase in glomerular hydrostatic pressure. When glomerular hydrostatic pressure increasing, there is an increase in glomerular filtration pressure. When there's an increase in filtration pressure, there's a more filtrate which is formed. The more filtrate, then there is more, the urine is hypotonic. Hypotonic. The next way is by decreasing the water absorption. By decreasing the water absorption. Edric 2 minutes absorption. Yeah. Please write down this first, everybody. Glomerular hydrostatic pressure, glomerular filtration pressure. Can you see the screen? Glomerular hydrostatic pressure and glomerular. No? You cannot see the screen? Wait a minute. Now, what about now? Okay, write down. When by decreasing, see, hypotonic solution you can get by decreasing the water reabsorption. So increase in blood pressure volume. Increase in blood volume. Activates the osmo receptors. This stimulates hypothalamus. Hypothalamus further it decreases ADH release from pituitary. When there's a decrease in ADH, it's an antidiuretic hormone we have studied. When there's a decrease in ADH, ultimately it's lead to decrease in water reabsorption from DCT, and correcting that. When there is a decrease in water reabsorption, then the resulting is hypotonic urine, right? So when there's an increase in blood volume in the arteries, blood vessels, this activates the osmoreceptors. These receptors will go and activate the hypothalamus and tell the hypothalamus to decrease less of release of the rest of ADH hormone from the pituitary. Now this ADH hormone which is less in secretion will absorb less of water from the DCT. You know that DCT is under the influence of hormone. Now, when there is a less in reabsorption of water, then ultimately it leads to hypotonic urine. Clear? Any doubts? The aim is to produce hypotonic urine to remove excess of water from the blood. This can happen by decreasing the water reabsorption. How is this water reabsorption is decreased? By decrease in the release of ADH hormone, that is antidiuretic hormone. The function of the hormone is to absorb water. When this hormone is decreased, then there is no absorption of water, much absorption of water from the filtrate. So that the filtrate or the urine that is coming out is a hypotonic. Any doubts? This is the first condition. Are you all there? Is all clear? Okay. Everyone, now next condition is when the body fluid or there's excessive blood loss. When volume of body fluid decreases, or there's a decrease. No, actually it depends how much the body needs to excrete the water out of the body. Sometimes both activate, sometimes one gets activate. It depends on the how much water needs to be removed. Or due to excessive blood loss, or due to dehydration. The reason can be anything, vomiting, etc. How can we restore the water in the body? That's a question. First one is by decreasing the ultrafiltration. By decreasing the ultrafiltration. What is the aim? Aim is to produce hypotonic or what is the aim? Hypertonic urine. Produce hypertonic. urine, right? So first method is by decreasing the ultrafiltration. So how is that done? Decrease in blood volume is sensed by decrease in renal pressure, decrease in renal arteries. There is a constriction in the wall of there. This leads to, when there's a decrease in the pressure, this leads to constriction of the wall of the arteries. There is constriction in the wall of artery or arteriole. It's called myogenic control. That is myogenic control. Then, when there's myogenic control, this leads to decreased flow of blood. Decrease flow of blood. So, decrease in filtrate or GFR. When GFR is decreased, there is hypertonic urine, less of water. This is how you can decrease. Myogenic controls gets activate when do you think the myogenic get activate? It gets activate when the blood volume and the blood pressure is decreased. Myogenic control, it is construction of the aim of the myogenic control is to decrease the diameter of the arteriole so that the less blood can flow. If the less blood is flowing through, then the less filtrate is formed. That less filtrate will give to hypertonic urine. GFR is less. Next, write down and do let me know once it is done. Next. Next method, first method is by decreasing the ultrafiltration. Next method is by increasing Water, exactly opposite, increasing water reabsorption, or by increasing release of which hormone? Increased release of which hormone? Which hormone? ADH. Very nice. ADH. So whenever there is decrease in blood volume, this leads to this will switch off osmoregulator. Osmo receptor. This will switch off osmo receptor. Then this leads to there is increase ADH release. So as we'll study the renin. Okay, that is a special mechanism. We'll study about this. Then there is an increase in ADH release. This increases the water reabsorption, and leads to hypertonic urine. Give me time, so as we'll read about renin. Very important about the renin. Did you all understood this? Any difficulties? Okay. Please write down this. The C1 is by activation of RAS. R is renin, angino, and aldosterone system. Very important, this system. Please concentrate. We'll write down first and then I'll explain you. It will be more easy. This order is very important. You need to remember that in this same order, renin, angiotensin, aldosterone system, the full form of RAS. Okay. The apparatus that uses juxtaglomerular apparatus, or the structure that is used here is juxtaglomerular. I'll let you know what it is. Glomerular apparatus. That is JGA structure. The special cells are called juxtaglomerular cells. These are the modified cells. These are modified. We'll write down and explain, okay? Modified cells present in the afferent arteriole. Present in the afferent arteriole, which stores inactive renin, which stores inactive renin. Please write down. Yeah, there's a difference between renin, this renin, and that renin. This renin is a different than that renin. That renin is double N ion, which is present in the infant milk. Is it clear? That renin is different. That is a digestion which is present in the child, right? And this renin is different. Please make a note. Can you see the screen? Can you see the screen? Now you can see. No? Okay. Now. I hope you can see. Okay, are you done writing this? Then we can move ahead with the renin. Now, RAS means renin, angiotensin, aldosterone system. When does this get activated? Let us write down that and then we'll discuss. Okay. RAS gets very important activated when blood, which leads to decrease by J juxtaglomerular cells or the apparatus. When they detect this, the juxtaglomerular cells screen is not visible. Is it screen not visible? Suniti, can you please re-log in and come? Release enzyme. Suniti, re-log in and come. Re-log in and join. Then this renin enters in blood. Enter in blood. When it enters in the blood, it will act on angio, let me write down, angiotensinogen. Angiotensinogen. It will act on angiotensinogen to give angiotensin 1. Angiotensin 1. Angiotensinogen, it is a plasma protein synthesized in liver. It is made of 453 amino acids, right? Then angiotensin 1, it is a decapeptide, that is 10 amino acid structure. This angiotensin 1 gets converted to form angiotensin 2. Angiotensin 2. It's a hormone of 8 amino acid. It's a hormone of 8 amino acid. Now, which enzyme will act here? The enzyme that acts here is angiotensin converting enzyme. Angiotensin converting enzyme from lungs or alveoli. From alveoli. Now, when there's a decrease in blood pressure, there's a decrease in blood volume, there's a decrease in blood pressure, will lead to decrease in GFR. When the GFR decreases, this is detected by the juxtaglomerular cells that release renin. Renin is an enzyme. Now, from the in size, tensinogen is a plasma protein that get converted into angiotensin 1, which is of 10 amino acid. Further, angiotensin converting enzyme from the alveoli will convert angiotensin 1 into angiotensin 2, which is a hormone. Now, this hormone plays an important role, which is of 8 amino acid. Is this point clear? Is it point clear? Okay, very good. Now, so how it's going to act on different system to form a proper urine, right? When there's a decrease in urine, how can you get a proper control on the water? So when the blood volume is lesser, the blood pressure also decreases. So the first one is stimulate adrenal cortex. Which hormone does the adrenal cortex? The adrenal gland of cortex. You know that adrenal gland has adrenal medulla and adrenal cortex. So adrenal cortex secretes an hormone or the release some hormone. Which is that hormone? increases release of aldosterone. What about aldosterone? Very good, Chrisley. Aldosterone. This will increases sodium reabsorption. Aldosterone increases sodium reabsorption from DCT from from DCT. Now, when the when the sodium reabsorption is increased, this will lead to increase in water reabsorption from DCT. This will increases water reabsorption from DCT. This is one way. So when the water is reabsorbed, a lot of hypertonic urine is produced. When the water is reabsorbed, what is the result? Formation of hypertonic. When the water is reabsorbed, lot of sodium is reabsorbed. When sodium is reabsorbed, there is a reabsorption of water, and that leads to formation of hypertonic urine. That is the aim of the RAS, to form hypertonic urine. The other way or the other method is constriction act on smooth muscles and cause vasoconstriction. So that when the blood vessels become narrower, so that the BP is normal. Because even the BP is low. If angiotensin act on the smooth muscle, it brings about the contraction and it decreases the diameter of the arteries so that the blood pressure becomes normal. The third is it will act on stimulates hypothalamus. Stimulates hypothalamus. Hypothalamus has centers for what? That increases thirst, and when thirst increases, there's a lot of intake of water. This is how you can get back the normal blood volume with the help of RAS. Do let me know if there's any difficulties. Doubts? It's clear. Others? Pratyusha has no doubts. Okay, very good. What about others? Is this everything clear? The new students who have joined? Any doubts? Okay. So if there are no doubts, we'll end the class for today. See you all in the next class. Next class will continue with the same. Once there's activation of RAS needs to get inactivate at some point, right? It cannot remain to be active all the time. So this part we'll study in the next class. Clear to all? Can we wind up the class if there's no doubt? Is there no doubts? Okay. See you all. Bye bye. Take care. You're welcome. Bye bye.